Hello students, welcome to today's lecture. It's about RNA. Okay, we are done with the DNA, its structure and then its synthesis. Now going to RNA. First of all, its structure. RNA, its full form is ribonucleic acid, okay, and it's made up of three components. Number one, there's a nucleobase or a nitrogenous base like we had in DNA. Here also we are having a nitrogenous base or a nucleobase. Then we are having here a ribose sugar attached to this nucleobase, and in DNA it was a deoxyribose sugar, but here we are having a ribose sugar, and then finally we are having a ribose sugar attached to phosphate group. Okay, and the discovery of RNA, it began with the discovery of nucleic acids by Friedrich Mescher in 1868. It was Friedrich Mescher in 1868 who discovered nucleic acids in the nucleus, and it was DNA plus RNA, but he was not able to say which is which. And since it was found in the nucleus, he named it as nuclein. Okay, now firstly coming to its first component, that is nitrogenous base. The nitrogenous base, it's as you have seen in the structure of DNA, it can be a purine or it can be a pyrimidine. If we will talk of purine, as you know, it's a two-ringed structure, and its numbering we say from this number 1, 2, 3, 4, 5, 6, 7, 8, 9. It goes against the Huckel's rule, but it's like it was given earlier to it. The numbering was given earlier to it, and it is just as a tradition we are following the same numbering. And remember, purine, it's two-ringed structure, and then we are having a pyrimidine. In pyrimidine we are having a single ringed structure, and the numbering is like this: 1, 2, 3, 4, 5, 6. Okay, now if we will talk of purines, we are having in RNA we are having two types of purines. Like first is adenine. Okay, adenine is formed simply by adding amino group to the 6th position of the purine structure. If we are having the purine structure over here, and it's the 6th position, at 6th position we will have attached amino group, and that will be formed adenine. So how we will name adenine? It's simply 6-amino purine. Okay, now another is guanine. Okay, another is guanine. In guanine, what is there is that at 6th position of this purine structure we are having a OX group, and at 2nd position we are having a amino group. So we will name guanine as 2-amino 6-oxypurine. Okay, now these are the similar bases which are formed in DNA. Here in RNA also we are having adenine and guanine. Now coming to pyrimidine. First pyrimidine is cytosine, okay, which you know it's also formed in the DNA. Cytosine is formed by attachment of amino group to the 4th position and OX group to the 2nd position. So its name will become 2-oxy-4-amino pyrimidine. Fine. And then lies the difference with the DNA. Instead of this thymine, here we are having a uracil. Okay, here we are having a uracil as a pyrimidine, as one of the pyrimidines we are having uracil. See, here pyrimidine is formed, uracil is formed by addition of OX group at 4th position and OX group at 2nd position. So we will name it as 2 and 4 dioxypyrimidine. And this uracil, it is different in RNA. Okay, there in DNA we are having thymine, and here in RNA we are having uracil instead of thymine. Rest is same, rest nitrogenous bases are same. Now the question lies that why there is uracil in place of thymine in RNA. Well, in early world, as we know, the genetic material when the world just was evolving many billions of years ago, it was RNA as the genetic material and not as the DNA. But the problem with those microorganisms was that RNA was the genetic material, but it was full of errors. It used to have full of errors, and that posed a problem to it from moving from generation to generation. It means from generation to generation, information used to get mistransmitted. Okay, and one of the errors was there the cytosine in RNA, it used to get deaminated to form uracil. Okay, it used to get deaminated into uracil. Now the RNA in RNA repair enzymes, they were confused that is it the uracil which is exactly at its position, or it is the uracil which has got incorporated there because of the deamination of cytosine. So the enzymes were not able to distinguish between the uracil, means should the uracil be there, or it is the uracil which has incorporated there at a wrong position instead of cytosine. So there was a problem in its correction. Okay, eventually the organisms, they chose the DNA as this genetic material. Here you can see cytosine, it upon deamination and addition of water, it changes into uracil. Okay, here we are having this amino group, and here we are having this oxo group. Upon deamination and addition of water, we are having two ox groups, and which forms uracil. So cytosine, upon deamination, it forms uracil. Okay, fine. Now, so organisms evolved their genetic material from RNA to DNA. Had DNA not switched from uracil to thymine, the deamination damage to cytosine would be essentially impossible to detect. But since thymine is used by DNA, if now uracil in DNA, if now cytosine in DNA will get deaminated to form uracil, okay, the enzymes will abruptly know that we shouldn't have the uracil, and they will remove it. Okay, that is why DNA over a course of time has chosen thymine over uracil as the genetic material. Having done with this nitrogenous bases, now another component of these ribonucleotides is ribose sugar. Okay, here also lies the difference. See, in DNA we were having this deoxy ribose. Here there was not this O group, it was simply H group, means oxygen was removed, so the sugar was deoxyribose. But in RNA we are having a ribose sugar which is having this 2' oxygen intact. Okay, and this 2' oxygen being intact, it makes this RNA susceptible to alkali hydrolysis. See, when this bonding occurs, okay, this bond, this OH, it forms a bond with nitrogenous bases. Okay, so oxygen is not available here. Then this 3' OH group, it forms bond with another nucleotide, forming 3' 5' phosphodiester bond. So this is again not there, but this OH remains available, and as a matter of fact this OH remains available, and as a result of this this RNA, it is susceptible to alkali hydrolysis. Okay, this is reactive because of this oxygen over here. And lastly we are having a phosphate group. Okay, so a ribonucleotide in this RNA will look like, we are having a nitrogenous base here. Here we have shown purine. It can be pyrimidine as well. Okay, then this nitrogenous base, it forms a bond with the ribose sugar, and the ribose sugar forms a bond with a phosphate group, and this forms a nucleotide, a ribonucleotide. Remember, we are having nitrogenous base, can be here adenine, guanine, cytosine, or uracil. Okay, and then we are having changes in sugar also. It's a ribose sugar here having 2' OH group intact. Okay, and the structure of RNA, it was given by R.W. Hawley in 1965. Now the chemical structure of RNA. Okay, see, this is one unit, one base, one sugar, and one phosphate. Okay, it joins hands with another base, another sugar, and another phosphate. Okay, this is a one ribonucleotide, and this is another ribonucleotide. See here, we are having 3' end of this sugar. Okay, 3' end of this sugar, it will form bond with the 5' phosphate of another that will be 5' 3' phosphodiester bond. 5' 3' phosphod, it is 3' of this and it is 5' of this. So 5' 3' phosphodiester bond. Also, as I have already taught in the class about this, that in bases we are having the simple numbering 1 2 3 4 5 6 in order to distinguish the atoms of this base from that of sugar. In sugar we use 1', 2', 3', 4'. Okay, that is what I mean by 3' and that is what I mean by 5'. Okay, one ribonucleotide joins hands with another ribonucleotide and one gives 3' and another gives 5' hand. So a bond is formed which we call as 5' 3' phosphodiester bond, and as a result of this we are having a linear chain in RNA is formed. Okay, and this structure, as I've already told, it was given by R.W. Holley in 1965. Okay, another important thing about this RNA, it is that it is single-stranded. Okay, in organisms, in most of the organisms, it's found in single-stranded form, except in some viruses. Okay, except in some like in rotaviruses, we are having a double-stranded RNA. Leave that, those are exceptions, but it is just a single-stranded RNA. Okay, it's a single-stranded RNA. Fine. Now, if you will see sometimes the structure of RNA like this, okay, it is like this RNA folds upon itself and the complementary base pairing, it creates a hairpin loop-like structure. Otherwise, this is also a single-stranded, this RNA molecule which you are looking here, it's also single-stranded. But only thing is that it has folded upon itself, finding the complementary base pairing. For example, if you will see here, if it is a cytosine, okay, and if it will fold upon itself, it will find a guanine and will bond pair it. But it is actually a single-stranded RNA molecule which has formed a hairpin loop-like structure and has folded upon itself. But mostly it is single-stranded. Okay, fine. This is one point about this. And another point is that it does not follow Chargaff's rule. Okay, what is Chargaff's rule? Chargaff's rule says amount of adenine equals to that of thymine and amount of guanine equals to that of cytosine. In other words, we can say amount of purines equals to that of pyrimidines. But here, since it's single-stranded, there is no complementary base pairing like we are having in DNA. There is a complementary base pairing. Okay, adenine always pairs with thymine and the cytosine always pairs with guanine, or we can say a purine always pairs with a pyrimidine. So the number of purines is equivalent to the number of pyrimidines. But here we are having a single-stranded DNA and there is no complementary fashion, and as a matter of fact we are having Chargaff's rule is not followed here. That amount of purines is not equivalent to the amount of pyrimidines. Okay, another thing is that from this RNA, it is synthesized mostly in most of the organisms. It's synthesized from the DNA. Okay, and the process of its synthesis from the DNA, it's known as transcription, which we will take in next lectures. Now coming briefly to types of RNA. Firstly, we are having a messenger RNA. This messenger of RNA, it is just a simple nucleotide sequence. Okay, it's just a simple nucleotide sequence. And here it just carries the instructions for assembling amino acids into proteins, which we will see later on in detail. And then we are having a ribosomal rRNA. It helps in the assembly of these ribosomes during the protein synthesis. We will take up this also in detail. And then we are having a transfer RNA molecule. This transfer of RNA molecule, as you can see, it's a structure like this. It's a clover leaf-like structure. Its structure we will take up in detail. Its structure we will take up in detail. Okay, it is also, you can see here, it has Also, it's also single-stranded, but it has also formed these loops due to complementary base pairing. Okay, there are no double strands here. It's just there's complementary base pairing, and it has formed this clover leaf-like structure. And during protein construction, transfer tRNA transfers each amino acid to the ribosome. We will have a look at these in detail later on. Now, another important thing about this is that RNA can be identified by Orcinol Colour reaction, or which we call as Bial's test. Okay, as you remember, I've told you that in RNA we are having a ribose sugar which is having an OH group. Okay, and Orcinol, it's a chemical compound. It reacts with the ribose groups in the backbone of the RNA molecule and depends on the formation of furfural. When the pentose is heated with concentrated hydrochloric acid, Orcinol reacts with furfural in the presence of ferric chloride and acts as a catalyst to give a green color. Okay, so ribose, whenever we add the ribose to this RNA mixture, it reacts with the ribose sugar, okay, forming a furfural, and that furfural in presence of ferric chloride, it gives a green color. Okay, so if in nucleic acid mixture we have to see whether there is a DNA or RNA, what we will do, we will add Orcinol to it, and if blue color is formed, it means RNA is there. If not, it means that it's only DNA. Okay, why? Because if blue color is formed, it means the reaction has taken place. It means that there is a ribose group. Okay, deoxyribose is not able to show this color reaction. This is the test tube which shows before this reaction. Okay, before this but upon adding the Orcinol and the completion of the reaction, we get this blue color, blue-green formation of color. Okay, now coming to the summary, what we have learned. RNA is single-stranded except in some viruses like rotavirus with double strand, which is having double-stranded RNA. It's made up of ribonucleotides with a nitrogenous base, ribose sugar, and a phosphate group. The nucleotides are joined together to form a polyribonucleotide 5' 3' phosphodiester bond. Nitrogenous bases are adenine, guanine, cytosine, and uracil. Chergaff's rule is not obeyed. RNA is susceptible to alkali hydrolysis. RNA can be histologically identified by Orcinol Colour reaction. RNA has a role in storage, transmission of information, catalysis, and the synthesis of RNA occurs mostly from DNA, and the process is known as transcription. This is all about this. Thank you.